Hi, my name is Ryan McNamara, and I'll be presenting this talk on repurposing exosomes for nanomedicine on behalf of myself and Professor Dirk Dittmer at the University of North Carolina at Chapel Hill School of Medicine. The problem that we are seeking to address is how can we safely deliver therapeutic agents? Nanoparticles can act as delivery agents of chemotherapies, and I tend to think of nanoparticles as these microscopic Trojan horses that can package and deliver these functional materials, such as drugs. Now, the pros to using delivery vehicles for drugs is that it can decrease the drug toxicity, it can direct the drug or the therapy to a particular cell type or specific tissues, and it can also preserve and stabilize the function of the encased drugs or materials. The solution that we're proposing is that this system of nanoparticle-based delivery has been mimicked in our bodies for as long as we have been around. And we do this through what are called exosomes. And we call exosomes as nature's own nanoparticles. So exosomes are small extracellular messaging vehicles that are released by just about every cell type. The contents of an exosome are lipid membrane-associated proteins, as well as encased proteins, as well as encased nucleic acids. In the middle, you will see a scheme of what a typical exosome would look like. And on the right, you will see an electron micrograph image of affinity purified exosomes that we took here at UNC. So how do we purify exosomes, and how can we repurpose them for nanomedicine? The solution that we're proposing is the production and delivery of genetically engineered exosomes. We have developed a high throughput method for GMP-grade isolation of exosomes from bulk fluids, such as human plasma or cell culture supernatant. We do this using tangential flow filtration using the scheme that you can see on the upper right. The exosomes can be custom loaded, such as the fusion of fluorescent proteins onto membrane-associated proteins on the exosome, such as an M-cherry, and what you see on the lower right corner of your screen is the exosome pellet from cells with that fusion protein. They can also be modified after you purify them by changing the internal cargo. And lastly, these exosomes can be safely delivered intravenously into non-human primates. So what you see here is an endothelial vein histology of non-human primates that were infused with exosomes that had the M-cherry that I just showed you on the previous slide. And what you can see here is the endothelial cells, marked in blue with their DAPI, uptook these exosomes marked in red. The market for exosomes is very similar to that of lipid nanoparticles. Nanoparticles deliver therapies against cancers and infectious diseases, showing that there is broad applicability in the sphere of medicine. The examples of nanoparticle-based delivery, particularly recently, would be the Moderna and BioNTech Pfizer COVID-19 vaccines, which are mRNAs delivered through a lipid nanoparticle. Other examples include Doxil, which is doxorubicin inside of a lipid nanoparticle, and Lipusu, which is paclitaxel inside of a lipid nanoparticle. All of the above examples have cases of reported allergic reactions and or anaphylactic shock. The advantage to exosomes is that they are nature's nanoparticles. So therefore, immune responses towards them would be much less than even artificially produced lipid nanoparticles. And on the far right, what you'll see is a PubMed citation index of exosomes, in which you can see that this is an exponentially growing field. The team responsible for the data that you've seen here is headquartered here at the University of North Carolina, but we also have collaborators at the Tulane National Primate Research Center, as well as NC Central University. I'd like to thank you for your time. You can contact either Professor Dittmer or myself using the contact information on the left, and I thank you for your time.